Assalamualaikum and a good day to all the panels. My name is Wanur Izrina binti Wan Izani, a final year student of Bachelor of Science Biomolecular Science, and I'll be presenting on my final year project thesis, Mode 1, entitled Comparative Separation Methods of Metabolomic Profiling of Human Milk Using NMR Spectroscopy. According to Samuel et al. 2020, human milk is a biologically complex and dynamic fluid that contains a wide range of nourishing substances, such as lipids, proteins, carbohydrates, fatty acids, amino acids, minerals, vitamins, and trace elements. Meanwhile, metabolomics is the study of small molecules called metabolites in a biological system. It aims to analyze and measure these metabolites to understand an organism's metabolic state and health. Human milk metabolomics studies the small molecules or metabolites present in the human milk. This aims to analyze and measure these metabolites to understand the nutritional and health benefits of human milk, as well as the metabolic processes that occur in both the mother and the infant. Moving on to the objectives of this research. The first objective is to determine specific metabolites present in the human milk using the Blythe and Dyer separation, which uses the methanol chloroform mixture method of NMR spectroscopy. The second objective is to identify differences in human milk metabolites using different separation methods by comparing to the other literature research. Next, methodology of the study. After receiving the ethical approval, the participants were selected based on inclusion criteria such as healthy mother without any medical history, 18 to 39 years old, and full-term birth. After that, human milk samples were collected from each participant during the lactation period from 0 to 6 months. 1 to 2 ounces of samples were collected and transferred to the microcentrifuge tubes before being stored in a freezer at -20 degrees Celsius. Next, sample preparation was made by using the Blythe and Dyer extraction method, which uses methanol chloroform mixture. The human milk metabolite extracts that were collected were then analyzed using 1H NMR spectroscopy. The data received was analyzed using TopSpin software and compared with other research that using the methanol water extraction method. This is the sample preparation method that I use, which involves a methanol and chloroform mixture. It is a mixture of two solvents typically used in a 2:1 ratio of methanol and chloroform. Methanol is a polar solvent while chloroform is less polar. Adding a methanol chloroform mixture to human milk facilitates the separation of polar and non-polar metabolites. Upon mixing and centrifugation, the solution separates into an upper aqueous layer containing polar metabolites like amino acids and sugars, and lower organic layer containing non-polar metabolites such as lipids and fatty acids. This biphasic extraction method enhances the subsequent analysis by isolating different types of metabolites, making it easier to study the complex biochemical composition of human milk. In Figure 2, A, B, and C represents the spectrum of 1H NMR spectroscopy of 3 samples of human milk from 3 different mothers. In NMR spectroscopy, the chemical shift is a measure of the resonance frequency of a nucleus relative to a standard. This measure provides valuable information about the electronic environment surrounding the nucleus. When identifying metabolites, the chemical shift helps determine the types of chemical groups present and their position within the molecule. For example, a chemical shift of 1.477 ppm was identified as alanine. Table 1 shows the 21 possible metabolites from samples and characterized them. A mixture of methanol chloroform was used for the extraction method. Identification of the metabolites was done by comparing the chemical shift with the literature studies from Fatih et al. 2017, Liang et al. 2017, and the Sunderkild et al. 2013. Thus, the objective 1 was achieved by determining the specific metabolites present in a human milk using Blythe and Dyer method. Next, Table 2 shows the 36 metabolites obtained from the literature that use the methanol water extraction method, commonly used when focusing on polar metabolites. Meanwhile, methanol chloroform method used when comprehensive metabolite extraction is needed, including both polar and non-polar compounds. Table 3 shows a combination of 48 metabolites detected by both methods. The metabolites were compared and listed down in the table. For sugars, Blythe and Dyer methods identify glucose and lactose from the human milk samples. Glutamate, isoleucine, phenylalanine, taurine, lactate, and choline were also identified. Other metabolites may not be detected, such as galactose, due to some human error during the experiments. Table 4 shows 8 metabolites that present and share the same metabolites for both separation methods. 7 out of 10 of the metabolites are polar metabolites. However, there is only one non-polar metabolite detected, which is isoleucine. As mentioned before, methanol water extraction method used when focusing on polar metabolites. Thus, the methanol water method is not suitable for detecting isoleucine. Isoleucine is a hydrophobic amino acid, meaning it is not soluble in polar solvents like methanol water. Ultimately, the methanol chloroform approach was selected for metabolomic profiling of human milk using NMR spectroscopy because it has greater capacity to separate a wider variety of metabolites, including lipophilic molecules, which are essential for a thorough investigation. Although the methanol water method is advantageous in terms of simplicity and safety, it is more suitable for isolating polar metabolites. On the other hand, the methanol chloroform method offers enhanced extraction efficiency and the ability to handle a wider range of metabolites, making it the preferred choice for comprehensive metabolomic profile. To optimize the efficiency of metabolomic profiling of human milk using the methanol chloroform method, various enhancements might be taken into account. An essential first procedure involves enhancing the extraction protocols through the manipulation of proportion of methanol and chloroform, fine-tuning extraction durations, and regulating temperatures to achieve the highest possible output and consistency in the extraction of metabolites.